The Aplysia californica is a mollusk that is known as the California sea hare. They primarily live along the coast of California. When they are threatened, they release a purple acidic ink that repels predators. In our lab, the Aplysia live in different tanks that are the same size. We monitor the temperature and the salinity of these tanks to make sure they are within the appropriate ranges at all times. They feed on red algae, which contributes to the coloration. Aplysia californica has the largest somatic neurons and growth cones in the animal kingdom. Their nervous system consists of 9 ganglia, 2 of which are buccal, 2 are cerebral, 2 are pleural, 2 are pedal, and 1 is abdominal. Their nervous system is composed of 20,000 neurons. Comparatively, the human brain is approximately 100 billion neurons. Because the Aplysia's neurons are so large, impulses can travel faster, leading to faster behavioral responses. Aplysia californica has been used as a model to study the neural basis of learning and memory because it presents 2 advantages for this kind of study: clear and easily elicited behaviors, and an accessible and relatively simple nervous system. This is why we chose this organism to study. We were introduced to classical conditioning by Pavlov with his experiments involving the dog. In this study, we used the same basic technique to observe the neural basis of learning and memory of the Aplysia. The Aplysia that we used for this study were split into 2 groups. One group was used for this protocol, and the other one was used for the reverse protocol, which we will explain later. The purpose of this protocol is to try to teach the Aplysia to respond to the brush in the same way that they respond to the needle, and to test the theory that they are able to learn even with such a simple nervous system. To do so, the first thing you do is touch the brush firmly against the tail of the Aplysia for 5 seconds, and then remove the brush and wait 10 seconds to see if there is a response. A full response would mean that the Aplysia's tail is completely withdrawn up into their body. We would record a full response, 3/4 response, 1/2 response, 1/4 response, or no response. If they do not fully respond to the brush, we would then poke the tail of the Aplysia with the needle and record and observe all responses to that. This can be done up to 5 times in one session with a minimum of 5 minutes in between each training. In this protocol, the unconditioned stimulus is the needle stick and the conditioned stimulus is the bottle brush. Here we have figures A and B showing the process of conditioning. Figure A shows the bottle brush being firmly held to the Aplysia's tail. Figure B shows the needle poke to the tail. These 3 figures are helpful in visualizing the responses of the Aplysia. Figure C would be no response because the Aplysia's tail is still visible and relaxed. Figure D is a full response with the tail not visible at all. Figure E is a full response with inking. This is a graph of our data for the conditioning protocol used. The Aplysia used for this protocol were labeled B and G. Aplysia B showed full responses during the first and second trial, and then later during the 14th, 15th, and 18th trial. Aplysia G didn't show a full response until the 6th trial. It showed full responses during the last 4 trials as well. The second group of Aplysia was conditioned with this protocol. The reverse protocol is similar, however we poked the Aplysia with the needle before we touched the brush to their tail. Aplysia A and C were used in the reverse conditioning model. They both showed mostly full or 1/2 responses to the unconditioned stimulus. The purpose of this material is to introduce students to the commonly used model organism, the Aplysia californica, and to study different aspects of learning and conditioning. Students learn the necessary conditions for classical conditioning and get hands-on experience with the laboratory activities later outlined in this presentation. To do a demonstration, you'll need the Aplysia californica, a tray, pipette, gloves, instant ocean, a bottle brush, and an 18-gauge needle with a 1 ml syringe. Once you have all your materials, you can begin the demonstration. To begin, place about 5 milliliters of instant ocean water on the tray. Place the Aplysia on the tray and then flatten it out gently with the palm of your hand so that the tail is visible and the Aplysia is relaxed. To demonstrate the unconditioned stimulus, obtain an 18-gauge needle and stick the Aplysia's tail with it. Next, observe the immediate response of the tail. The Aplysia may also ink. To demonstrate the conditioned stimulus, you want to do steps 1 and 2 again. Then take a new Aplysia or allow the one already used to rest for 5 minutes. Place the Aplysia on the tray and flatten it out so you can see its tail. Then gently touch the tail with the bristles of the bottle brush and observe their reactions. It is recommended that you try to get the Aplysia californica completely relaxed. A video demonstration may be best for a larger class size, and you want to practice with the bottle brush on your own hand so you can see how much pressure should be applied. This hands-on laboratory activity will focus on the importance of pairing the conditioned and unconditioned stimulus in the correct order. 4 groups are needed for this activity. Each group will need a full set of the materials that were used in the demonstration. Group 1 will focus on the conditioned stimulus only. Using the same technique as in the demonstration, they will record the Aplysia's reaction to the bottle brush touching their tail. Group 2 will focus on the unconditioned stimulus, the needle stick only. Group 3 will perform the unconditioned stimulus followed by the conditioned stimulus. This is the same protocol as the reverse conditioning explained earlier in the presentation. Finally, Group 4 will perform the conditioned stimulus followed by the unconditioned stimulus. This will be the same protocol outlined as the conditioning protocol. An example of a data sheet from the activity is included here. In Group 1, there should be no response, since it's a negative control group. The Aplysia californica in Group 2 should have the withdrawal response every time, since this is a positive control group. Sometimes there will be a stronger response with repeated needle sticks, but this doesn't always happen. For Group 3, there may be some association between the conditioned and unconditioned stimulus, but the conditioned stimuli is given after the unconditioned stimuli. The association will not be as strong as in Group 4. Group 4 is the group that should have the strongest association between the conditioned and unconditioned stimulus. The goal for Activity 2 is to learn how long it takes the Aplysia californica to learn and remember. For this activity, there will be 2 groups, a control group and a treatment group. The control group will only be exposed to the bottle brush. Their reactions will be recorded. The treatment group will experience the entire conditioning protocol. A sample data sheet is included here. Conditioning should occur in the treatment group, but not in the control group. However, the degree of learning and memory in the treatment group may vary. This is an example of the data from one of our studies. This proved to be an effective model for a small undergraduate or high school class setting. It is ideal for a minimal budget as the Aplysia are easy to care for. Multiple studies done by Dr. Hale and the undergraduate students at Quincy University have shown that this model is effective. The demonstration will help students visualize the conditioning process, and the activities will engage students in their work. The activities will also allow students to become more familiar with an organism that is utilized by researchers around the world. Finally, this method forces students to think critically.